Yeah, you guys come on around. Okay, so the equipment you see here is basically what you got to have lined up to do your blood type, all right? Prepare everything before you really start pricking anybody's finger, all right? I always see some students that are ready to grab the thing and just poke their finger, and then they're, oh, what do I do with my blood? I'm bleeding everywhere. Wait, okay? Get it lined up first before you start all that, all right? So first of all, you have a nice selection of weapons you can use. You can pick this one. You like that one, Chris? No. Or you can use the scissors. No. No. Oh, razor blade. Razor blade would work. No. Yeah. No, don't use any of that. Okay. That was joke. Joke. Okay. Delete the picture of that stuff. Okay.
I'll post that.
You can use the toothpick if you really want to. Just jab it in.
Got it.
Yeah, that's why you need 5 of them, right? One for each finger. Acupuncture.
That hurts.
All right. So anyway, you do need this stuff that you see here. There are 2 official kinds of lancets that you can use. Where are they? I'm trying to keep that finger clean because I'm going to do that one. Pass this around. Hold it up to the light and just see that there is a point at one end of the instrument that's in there. You can hold it up and see what I'm talking about. The lancets like this are perfectly usable. They are kind of our second choice because they tend to go deeper, all right? They tend to go deeper and make a larger puncture. So if you're having trouble getting a blood drop with the other technique, then you may have to go over to one of these, all right? Yesterday, I had to use one of these on myself, all right, to show the class, so I hope not today. It works just fine, but it's not preferable anymore, all right? I'm going to keep one out just in case I have to do it, all right? And you got to hold it like this so you can see where the point is. If you do end up using one of these, make sure you open the end opposite from the point, all right, so you don't touch the point. The point is sterile and dangerous too. All right, so our proper technique is going to be done with these things. Pass these around. You can open it up or grab 1 or 2 off your table. Get a couple from your table so you can practice with this while I'm talking. Yeah, give 2 or 3, every 2 or 3 people. Okay, these are called lancing devices. And they're going to be used with lancets, okay? Lancet tips or just lancets. These are the lancet tips that are going to go in there. Let's look at the device first. It is cocked, we say, by pulling it back, in this case. And then you shoot it with a button, all right? Now, some of them are newer, and then you just have it's just like a pen. It shoots and cocks at the same time, just like that, okay? But these are a couple years older. You pull it until it clicks and then shoot it, all right? Inside is where the lancet is going to go, all right? This thing is going to fit in there.
I thought it was a toothpick.
I thought it was a pen.
You have a lot of space between your teeth. That's a toothpick, yeah. Now, this is going to fit in there, and that will shoot out and back in again to create the puncture. On the outside, we have a scale, and that is a depth scale. All right, that's how deep it will go. So this one is right now set on the deepest setting, 5. All right. Women tend to have thinner skin, so women may get away with a 1 or 2 or 3. All right. Men tend to have to go to a higher number. All right. Personalized, it's personalized male, female thing. We tend to have thicker skin. It also depends on how much subdermal fat you have, all right? Okay, how much padding you got, all right? But for me, I generally always start with 5, okay? I use my hands a lot to work and everything, so I kind of have thick skin. But you can try it with a shallower setting if you want to. Now, big question everyone has. What do you think the first question everyone has about this is?
How deep we need to puncture in order to
okay.
What if the first one fail, right?
Boy, none of them. Your class is so unique. Nobody asks the normal. Most of the time, people ask first, does it hurt? Okay.
Cost.
And what?
Nothing.
You're not doing it, right? Okay, yeah, please don't try to do this yet. The answer to that question is it depends on how lucky you are, all right? Because the point of these is really, really small, all right? It's smaller than the end of the toothpick, all right? It's small enough that you can, if you're lucky, actually puncture between your pain sensors in your fingertip. And if you miss the pain sensors, you won't feel anything, all right? If you happen to hit a pain sensor, you'll feel a little bit, but it's a little bit, all right? It's actually a lot less pain than if somebody pinches you with their fingernails, all right? You know, so it's not much, really. Mostly students yesterday didn't feel a thing, all right? Mostly. So the answer is usually not.
How
Does it
unlucky are
hurt?
you,
Usually
right?
not, all right? The larger lancets, usually yes. It does hurt, all right? But not much, not for very long. Okay, so that's the lancing device and the lancet. They're in these boxes. Each table's got some of those around. Next thing, before we start lancing anything, we want to set up our area for collecting the blood. Use a clean slide. You can use new slides today, not the old ones you have to wash. Take a clean slide out, try to touch only the edges. All right, and create your half toothpicks. All right, how many half toothpicks does one person need?
3.
3 halves. Not 3.5. Line them up like that.
Oh, you're pretty strong.
Really? It's a toothpick.
It's a toothpick.
Can you break the end of that for me? Oh, he's so strong. Are you sterile?
No. What do you
No.
mean sterile? What do you mean sterile?
You just contaminated my toothpick with your fingertip. Okay, so there's a spare half toothpick. All right, you need 3 of those, and you need to line up your antibodies, all right? They come in these bottles, anti-A, anti-B, and anti-D. What's anti-D about? That's the name of the RH antibody, all right?
We're gonna have a blood test today.
Okay, so you put that on there.
Ah.
Just put it right on there. You don't put it in the toothpick, all right?
Okay.
Try to give yourself more space than that. I'm really trying not to touch it. Okay, whatever.
Let's just remove those.
All right.
That
Okay, so that's my anti-A, anti-B. And these are produced by a kind of biotechnology, all right? The gene for antibody B, all right, has been added to certain kinds of bacteria which are grown very rapidly in reactors, and then the bacteria express that gene and express the protein therefore. And by purifying it, we get unlimited amounts of antibody. And in the very old days, they had to extract that from people, all right? Blood donations were purified by other techniques.
So it's from bacteria, right?
It's
Also.
human antibody A or antibody B, but grown by bacterial action. All
Just
right.
like insulin nowadays, right?
Yeah,
Yeah,
insulin
insulin.
is one of the earliest ones. That actually goes back
It
to
used to be
like
very
the
expensive, but now it's quite
1960s.
cheap.
All right. Yeah, insulin can be grown cheaply nowadays. All right. My fingers are too big. I can't do this well. All right, just have those ready to go. All right. Okay, so you got them lined up. Now you're ready to do the prick.
Do you need
Set it on
any
5. I haven't finished sterilizing yet. At this point, take some of your ethanol, spray the end of that, all right? Many people share these things, all right? So spray the end of it a little bit, wave it around. While you're at it, spray the finger, which you're gonna prick, all right? Wave that around, don't touch it to
Can we
anything.
blow it to make it a little bit dry?
No.
Ah.
Of course not.
What do you think? Explain it. Explain it, if anybody else has that idea. Why is blowing on it a bad idea?
Blowing bacteria?
Putting bacteria
You're
into it.
putting bacteria right back on it again. Yeah, exactly. Okay, so you load it up like that. Actually, I cocked it by accident, all right? Okay, and then you twist off the cap.
So that's a cap.
That is a cap. And it's supposed to come off. Okay, that's a cap. Keep the cap. And there's the needle. It's very tiny. Okay, as I said, smaller than a toothpick by far, right? That's why it is possible to actually miss your pain sensors, right? Sometimes, if you're lucky. Right? Okay, put your cover back on there. All right, cock it like that. Okay, so it's ready to go. You notice when it works, you can't really see it working. Okay, it actually extends out a millimeter or less and pops back in again. And it all happens so fast you really can't see it. All right. Okay, so you're ready to go. Am I ready to go? Not quite. One thing I forgot. You need your hematocrit tube. Could you remove one tube from there? Don't touch the other end. Don't touch the other end. Touch only the end that has color on it. I almost forgot that. You need that ready as well. Okay. The colored end is for holding, the other end's for your blood. All right. Okay, am I cocked? Okay, I'm now cocked. I'm gonna set on 5. Lay it on there. Push down hard. Now this is where most people make a mistake. They don't push hard enough. Okay, and I got a little tiny blood. I felt nothing. I seriously felt absolutely nothing just then. Keep that in case you do have to do it again. And I didn't get much. That is not really enough. That is not going to work. That's not enough. Now, to get the blood out, you kind of massage and let it flow.
Maybe you should remove the ring.
Yeah, I think the ring is causing that problem, huh? Well, it's okay. It's good for a demo. Okay, so that was a failure. I'm going to use the other way. I'm going to use this one. All right. Okay, it's all right. It's all right. I've done this 2 or 3 times a year for 18 years. All right, so that is still very sterile. And I'm just going to, again, hold it on there fairly hard. Put that against that point and stab it, okay? And that one should be working. Yes, there we go. All right. Okay.
So your body
How much do you
develop
need?
thick skin, right?
Yeah, I've got too thick skin. Excessively thick skin. Now, hold this only by the blue or red end, depending on which bottle you got. Get a nice drop of blood out. As I said, you squeeze a little bit and let the blood flow, and then squeeze it again, let it flow. And put the capillary tube right there, and you'll start getting some blood, okay? You need about 1/2 a centimeter to 1 centimeter, all right? Is good,
So about
okay?
5 pounds for
Something
half a
like
kilo.
that, yeah. It depends how easily you bleed. Whoops. Don't touch it. If you touch it, then you're possibly causing a sterility problem.
How to do it now?
Okay. Okay. And this is exactly the same spot I did yesterday for your friends. Maybe that's why it's not wanting to bleed anymore. Body's saying once a term is enough. And this is 2nd time in 2 days.
Next time use the video.
Oh, really? That's not as good, is it? We prefer live teaching, don't we? Not virtual teaching. Okay. I'm not getting much out, that's for sure.
Wait, so we need half of it?
Half.
5 millimeters would be good. Okay, now one thing about these tubes, you can pass that around, you can read it. It mentions a substance called heparin. Okay, I just got some more. Heparin is a molecule that prevents blood clotting. Do remember, and take a note if you have to, that clotting is different than agglutination. The process you'll see in the blood reaction is called agglutination, all right? Blood clotting is done with the platelets, okay, and the red cells usually. And that's what's happening inside my skin right now. It's clotting, right? And that's why it's not flowing very much anymore. Okay. But as long as I have the heparin-coated tube, it will stay liquid in there, all right, for quite a while. All right, now one other thing I didn't prepare was a cotton ball, or plaster, yes. So if you look on one of the other tables, you might see a little, yeah, gauze. Okay, can we take one of those out? I think we're kind of running out of this. All right. Nurse Mabel, could you get the scissors out of there and cut that in half? I don't need that much. They're not sterile now. Just better than nothing. Okay. Let's do something to staunch the bleeding a little bit. Don't lose your sample. All right, now. That's why maybe you need somebody else to help you with this, all right? Okay, now tap out. This is where you have to work fairly fast. Tap out a small amount of blood onto the, oh my God. It's not even coming out. Oh, gee, this is not working as well as yesterday. I was too slow. It's too thick. Ah, this is not working at all. What's going on here? I got to do it again.
Could you blow it?
No, don't blow it. Then it'd all come out.
No, if you blow it, you can blow it.
Oh, I was too slow. It dried out.
Do it again.
Oh.
Maybe you can try with a finger that you didn't wear a ring with it.
Definitely.
You need a volunteer.
Okay, I'm gonna do it again. Do it again.
Okay.
I need a new tube. Yep. Yeah. Why not? Okay. I don't like this, but I'm gonna do it anyway. Jesus. It definitely hurts. All right. Okay. One more time. Do it faster. You know, believe it or not, for a couple years I got really tired of doing this, so I asked students for volunteers. Now I'm back doing it again, so I guess you guys are lucky, right?
You can just do it the video.
No.
One drop from each. Yeah. It's
Okay.
good now.
Okay.
You don't mind the
Oh
I don't know what's wrong with me. I'm definitely not in good shape today. All right.
That's why you told us
Okay,
that there's going to
sorry
be
about
blood
that.
bleeding around.
I have to wash my clothes, I guess.
Was that blood?
Yes, your blood.
Oh, jeez. Oh, I'm sorry. Oh, jeez. I'm really sorry.
Maybe you
okay,
use
you gotta wash it off. Yeah.
peroxide.
Yeah. Put a little alcohol. Okay. Spread some alcohol on that. It'll help keep it from staining.
Maybe you need hydrogen peroxide.
Oh gosh. Okay. I don't know. I'm sorry. I'm really sorry. I don't know what I'm doing wrong today. I must be in a hurry or something. Normally, okay. You tap it out. Tap it out. Say it got some blood there next to the drop, all right, and then you mix it up with your toothpick. Okay, one toothpick per blood drop, all right, like that. Don't mix the different kinds together, all right. Mix it in quickly, and then you will see agglutination or not. All right. Now the one thing to be aware of is that the D takes a long time to react. All right, it takes a very long time to react. It can take 2 or 3, 4 minutes. The other two react pretty quickly. So if you're A or B or O, you know that pretty quickly. But the other one, after you've left it in there for 2 or 3 minutes, shake it. And that often will get the red cells to agglutinate. All right.
Why does it take longer?
It has to do with the size of the antibodies, as far as I know, all right, and the number of them. I think the number of anti-RH that you have is not very big, so it takes a while to react. But the agglutination is very obvious. It'll look like little spots, little dark spots in there. If it doesn't agglutinate, it'll be a smooth, dark red or even black brown color, all right? No spots. Okay, go for it.